We're going to continue going through the dynamics of DNA replication. Remember that this process is really just simple pieces, but it's a few simple pieces, and that's what makes it a more complicated situation. Like, for instance, what's the enzyme that causes this melting of DNA here? Well, that would be DNA helicase. It's not shown, but it is here. Or the enzyme that's involved in relieving tension in the DNA. Well, we call that topoisomerase, or the single-strand binding proteins. And now we're going to get into RNA polymerase, like for instance, RNA polymerase 3 is synthesizing from the 5' towards the 3' end, reading 3' to 5'. And the opposite is going on, and it's going on the opposite one, but again, we're synthesizing 5 to 3. So we started here, and then we synthesize here, and then we'll synthesize another one, and so forth. Always reading 3 to 5, synthesizing 5 to 3. So let's pick this up. So reading 3 to 5, you would know that this is the leading strand, right? So we have our primase that created our RNA primer, and now we have RNA polymerase 3 that comes in and adds a DNA nucleotide to that 3' end, synthesizing into the replication fork. So that's leading strand. That's the entire story. As helicase unwinds, RNA polymerase 3 comes in, and it's going to displace those single-strand binding proteins as it synthesizes. So we've synthesized a little bit more, to the tune of about 50 nucleotides a second in eukaryotes and 500 nucleotides a second in bacteria and prokaryotes. So to do the lagging strand, we actually have to work away from the replication fork, and that means we have to have room. Here we have plenty of room to keep synthesizing. As long as the fork keeps opening up, we can keep synthesizing. In the case of the lagging strand, we have to open up enough room for, first of all, primase to fit in there and create a primer, and then polymerase 3 to fit in there in order to synthesize until it hits that previous primer. But it can't anneal DNA to RNA. It can't hook those two pieces together. And those pieces are known as fragments, or Okazaki fragments. So the part of the lagging strand synthesis we have to deal with is how are we going to get, first of all, get those primers out of there and then ligate the individual fragments. Well, an enzyme known as DNA ligase is one of the DNA repair genes that is involved, one of the DNA repair proteins that will assist in joining those fragments. But we can't go straight there. We have a couple of in-between steps we have to get to first. And so here's our leading strand going right in there, and here is an example of the lagging strand, and you can see the dynamics here. Now, ligase will repair these things, but it can't do it immediately. We have to get rid of those RNA primers. We can't ligate the RNA to the DNA. So we have to get those primers knocked out of there before we can do the annealing and the ligating of those Okazaki fragments. So now let's look at how lagging strand works. So replication fork is going to be up here. So we're leaving a lot out in this picture. So as enough space is created in the replication fork, primase will create that RNA primer, about 5 to 10 nucleotides. So that's primase there. Once that primer is created, DNA polymerase 3 can then synthesize, again, synthesizing 5' to 3' away from the replication fork. Okay, so we're leaving out the replication fork. We're just showing you the lagging strand. So we're moving, the replication fork is moving to the left, but we're synthesizing to the right, away from the replication fork in the lagging strand. And we are showing here the different pictures as time progresses. So this is earlier, and this is later, and this is later. So DNA polymerase III is synthesizing this way until we bump into that previous RNA primer. So we're synthesizing down, we bump into that primer, and we can't attach that DNA to the RNA primer. And so we have completed the creation of an Okazaki fragment. And DNA polymerase III falls off. But it leaves that nick area there between the DNA and the RNA primer previous. Now in this picture, what we've done is we have attached a primer using RNA primase. We've synthesized the RNA primer when we have enough room, and RNA and completed, and then DNA polymerase III has come in and created this fragment. So here's one for a reference, 1 and 1. So we're showing this over time. As more space is created, we are creating more Okazaki fragments. Now let's get to the next piece of the puzzle. See, now we have a different protein here. Up until now we had primase and polymerase III. Primase and polymerase III. Once we've completed this Okazaki fragment, another form of DNA polymerase comes on the scene, which is going to be involved in the repair and the ligation of the Okazaki fragments. So we have ligase, that's coming. But before we can use the ligase, we need DNA polymerase form 1. DNA polymerase 1. This is DNA polymerase 3. These orange ones are polymerase 3. This is primase up here. DNA polymerase 1 differs from DNA polymerase 3 in a very, very important way, in that DNA polymerase 1 can synthesize more nucleotides, and it can get rid of the RNA. It has what is known as exonuclease activity. RNA polymerase or DNA polymerase 3 does not have exonuclease activity. So DNA polymerase 1 can come in and commence synthesizing more DNA as it's popping those RNA nucleotides out. So as we pop out those RNA nucleotides and we get to the next DNA, then DNA polymerase 1 goes away. So it cannot ligate the DNA. It can remove the RNA primase. Let's say the old RNA. So it's removing the RNA primer. It's removing the RNA primer, and then once it gets to that DNA, it leaves, and RNA primase enzyme will come in and seal that phosphodiester bond in the backbone, in the phosphate. So we're going to fix that nick in the backbone. So that's called DNA ligase. So again, primase makes a primer. Polymerase 3 synthesizing 5 to 3 until it bumps into the previous primer right here. Then DNA polymerase 1 comes in, will remove the RNA nucleotides as it's synthesizing more of that Okazaki fragment until it bumps into DNA, and then it leaves. And then lastly, we have DNA ligase that will anneal the phosphate backbone to create one continuous sequence of DNA. So now let's show it all together. You've got your leading strand just chugging along here. Once it created the primer eons ago, that DNA polymerase 3 is free to just keep moving forward. We've got our helicase here. We've got our single-stranded binding proteins. All right, and here's our lagging strand. The only protein missing from this figure, the only protein missing from the figure that we've talked about is topoisomerase out here. It's the only one. So we've got our primase here that fit inside there's enough space in the replication fork that primase can come in, synthesize a few nucleotides of RNA. Then DNA polymerase 3 will eventually come in here and synthesize until it hits that primer. DNA polymerase 1 will then take over and with its exonuclease activity, remove those RNA nucleotides, synthesizing more DNA. And then lastly, DNA ligase will come in and anneal that phosphate backbone to create a continuous strand that looks a lot like the leading strand, only it's synthesizing away periodically as space is created in the replication fork. And so each replication fork has a leading strand. So it's showing in this box right here. Here's our leading strand. There's our lagging strand. If we were to slide this picture to the other replication fork, you'd see that this strand is now the leading strand again because we can synthesize 5 to 3 and read 3 to 5, and lagging strand on the other side. It's always about the orientation of the reading and the synthesizing. If you're synthesizing into the replication fork, it's leading strand. If you're synthesizing away from the replication fork, it's lagging. And in here, what I've done in this photo is add that topoisomerase enzyme. All right, so now we know where the topoisomerase is. It's out here in front of the replication fork, relieving the tension that's created as we unzip. The phrase is melt the DNA. So we've got that helicase here, single-strand binding proteins, and topoisomerase. They're involved in protecting and getting the DNA separated. DNA polymerase 3 chugging along into the replication fork on the leading strand. The primase, when there's enough room, comes in and creates a little RNA primer on the lagging strand to be followed by DNA polymerase 3, then DNA polymerase 1, and then ligase. So we slide down the strands a little bit, we can get a better picture of the lagging strand. So it's showing the box here as if we were seeing the replication fork, but you can clearly see it's not there. So actually what we're seeing is more like this rather than the whole replication fork on this side. In any case, we can see the RNA primer that was created by primase and now DNA polymerase 3 synthesizing the DNA until it bumps into that previous RNA primer. Once it does that, DNA polymerase 3 falls off, and DNA polymerase 1, with its exonuclease activity, enzymatic activity, in addition to the DNA synthetic activity that it has, displaces those RNA nucleotides, synthesizes additional DNA to fill those 5 to 10 nucleotides from the RNA. Once it bumps into the previous DNA fragment, it falls off and DNA ligase comes in and anneals that phosphate backbone. It's not adding any nucleotides. It's the hydrogen bonds that form between the bases, those form on their own. It's the phosphate backbone that DNA polymerase 3 and DNA polymerase 1 are involved with. They're displacing those nucleotides in DNA polymerase 1, those RNA nucleotides, excuse me, and ligase will come in and seal that final ligation of the phosphate backbone. So when we've been talking about this DNA replication, we've been talking as if these proteins kind of come and go. Well, that's not the whole story, actually. They're actually all sort of connected. They're all sort of connected loosely in what we call a DNA replication machine. It's actually a complex of all these different proteins that are associated with weak bonds. They're not super strong bonds, but nevertheless they are weak chemical bonds that hold everything together. And it turns out that we're not moving along the DNA. We're actually keeping the enzymes together, and we're actually reeling in the parental DNA and making new DNA from a relatively stationary DNA complex. Kind of like a fishing reel reels in the parental strand and extrudes, that's the phrase there, pushes out a completed set of daughter DNA molecules. And so here we show the entire setup. Maybe, I don't know if ligase is in here somewhere. I don't see it. But in any case, DNA polymerase 3 and helicase and so forth are all connected in this machinery that involves leading and lagging strands. It's a very, very amazing process how it works. And when we get to class, I will show you a video. It's hard to take in all the pieces in the video because it's happening so fast. That's why we break it out into individual pieces. But the important thing here is this has to work correctly or we get mistakes. If we get mistakes, we either have to repair those mistakes, which takes time and energy, or those mistakes get incorporated into the genomic language of the DNA and can lead to problems or unexpected results that are difficult to manage. Occasionally, very, very rarely, we'll have a mistake that fits in there and confers a benefit, but that's exceptionally rare. And speaking of repairing in DNA, that the DNA polymerases, especially DNA polymerase 1, can actually double check to make sure that the nucleotides are correctly matched with the opposite DNA sequence. So the DNA polymerase is actually proofread, so DNA polymerase 1 in particular, and if there's a mismatch of nitrogenous bases, these repair enzymes can come in and correct those things so that we're staying faithful to the original DNA sequence. DNA damage happens all the time, whether we're exposed to harmful chemicals, particularly with hazardous behaviors like smoking. And I'm not just talking about tobacco smoke. If you're inhaling burning vegetable matter, trust me, you are being exposed to certain chemicals. But these chemicals can come through different types of cooking methods, can be because you're out in the sun or around sources of radiation. And that's why folks who work in healthcare, especially around machinery that uses X-rays, have to monitor their exposure so that they aren't inducing excess DNA damage. But it can also happen just spontaneously. It happens every once in a while, and the enzymes are there to read and repair. There are two forms of DNA repair, and I really just want you to be aware of them. We have mismatch repair when you have a mismatched nucleotide, and you have excision repair, which is where like a DNA nuclease comes in and replaces a stretch of damaged DNA. So there are two different major mechanisms. So for instance, here's a example of excision repair. This right here, this little connected, you see there's a bond formed. This is usually happens between two T's. So if we have a T residue here and a T residue here, if you're exposed to UV light, like if you're out getting a tan, you're forming what are known as thymine dimers. That's where the two consecutive T nucleotide bases form a bond, and it can't be cut. So what happens is we excise a few nucleotides around that thymine dimer and replace it with new DNA. So we have a nuclease that will replace DNA polymerase 1 will come in, synthesize new DNA, and then ligase will come in after it. So we use a similar mechanism that we use in lagging strand in order to repair a very, very common error that occurs just by simple UV exposure out in sunlight. And if you remember in meiosis, we talked about genetic variability. Well, there is an input for genetic variability here in DNA replication. In eukaryotic DNA, the error rate is very, very low, but it's not zero, especially if you get lots and lots of errors. So if you are really into getting suntan all the time, you're generating so many errors that the likelihood of an error being incorporated into the DNA goes up. So it's low, but it's not zero. And some changes may become permanent. Now, if they're permanent in a somatic cell, they're permanent for that person. And that's where we can get things like moles or tumors. Those are basically caused by genetic changes in somatic cells. Once in a very great while, you'll have a mutation that occurs in a germ cell. The germ cells are the cells that make our gametes. They make sperm and eggs. And so if there's a mutation in a germ cell, and that one will get passed on into the next generation. So there is a very, very slow change in the DNA sequences over generations, which either get culled out or emphasized by natural selection over time. The best example I can think of for this in humans was during the bubonic plague in the what we called, which led to the dark ages of human existence, somewhere in around 900 to 1200 or so AD. We had bubonic plague broke out, and it literally altered the genetic sequences of humans in Europe, because people who had immune systems with a certain DNA sequence were susceptible, and others, a relative minority, that it had mutations in that, and thus had a slightly different immune makeup, survived, and they survived and reproduced, and so it changed the immune system profile of Europeans during that time, from before and after. We can examine bones and human remains, and we can see that process. But it's also super important in cancer. If you're exposed to mutagens, like for instance through smoking or certain types of occupations or certain types of diets, it's okay to have a modest diet. The problem is if you're eating it all the time, like for instance, well-cooked red meats have higher mutagens in them, especially if they have a lot of charring, if you know what I mean. So if you like your, hamburgers and steaks really well done, you actually have higher exposure to mutagenic compounds. If you like to suntan a lot, you're higher exposed to the mutagens of ultraviolet. Smoking, tobacco smoke and marijuana smoke are the two leading forms of inhalation smoke exposure. All will contribute to, first of all, inflammation, and that causes DNA cells to want to divide, and then when they replicate their DNA, then we get increased frequency of mutations. But we can also perhaps see some of this as well in other diseases like Parkinson's and other types of mutations that occur that seem to have arisen from DNA replication. DNA replication is an inherently mutagenic event, just because every once in a while there's an error in there. And so that completes this chapter. There's a lot going on mechanistically. You have your anatomy, if you will. Now we're missing topoisomerase in here. That's the one that's missing. Okay. Make sure that you can be able to identify all of these different enzymes and talk about what they do. What does DNA polymerase III do versus DNA polymerase I? DNA polymerase I has two activities, where DNA polymerase III only has one. But it's not enough to say just synthesize DNA. You can't just be that brief. You have to be more articulate. Moving into this field of biology and molecular biology, it's good to have an awareness, but for just a little while, you have to be just a little more articulate about what each and every one of these enzymes is doing. The helicase here is melting the DNA, separating the two DNA strands. Single-stranded binding proteins, holding those strands open so they don't reanneal behind the helicase. Topoisomerase out here. DNA polymerase 3 reading 3' to 5', synthesizing 5' to 3' DNA. Primase, RNA primer, and so forth and so forth. It's little simple pieces. Good practice for down the line when you're working in therapeutics. Therapeutics is very similar in that way. You're going to be thinking about several simple things, trying to get after a cause of disorder or injury or rehabilitation. So thank you for your time. One of the more technical chapters in this semester, and then we have one more chapter, and then we are complete. So thanks again, and I will see you soon in Chapter 17.